What if I told you that every time you concentrate, really concentrate, something physical happens inside your brain? Not a metaphor, not a motivational idea, an actual measurable biological event. The neurons in your prefrontal cortex begin to synchronize. A neurochemical called acetylcholine floods the synaptic gaps between cells. And in that window of focused attention, your brain literally becomes more plastic, more capable of rewiring itself. This is the neuroscience of focus, and it changes everything about how you should be working, studying, and living. My name is NeuroWise, and today we're going deep on one of the most powerful tools your brain has, and almost no one is using it correctly. We're going to cover what focus actually is at the neurological level, the three key neurochemicals that make or break your attention, how focused attention triggers neuroplasticity, and the specific science-backed protocol you can start using today. Let's begin. Most people think of focus as a mindset, something you either have or don't, a personality trait, a discipline problem. But neuroscience says it's none of those things. Focus, real deep focus, is a biological state. It's a specific pattern of neural activity, primarily organized by a region called the prefrontal cortex, working in coordination with subcortical structures like the basal ganglia and the thalamus. Here's the key concept. Attention is fundamentally a filter. Your brain receives somewhere in the range of 11 million bits of information per second from your environment. Your conscious mind can process roughly 50 of those bits. 50 out of 11 million. Focus is the neural mechanism that decides which 50 bits matter. It literally gates reality. The neuroscientist Michael Posner, one of the pioneers of attention research, described three separate networks in the brain that govern attention. The alerting network, which controls arousal and vigilance. The orienting network, which physically directs attention, including through eye movement. And the executive control network, centered in the prefrontal cortex, which manages top-down, goal-directed focus. Each network is distinct, each can be trained, and each responds to different neurochemical signals. That's where it gets interesting. There are three neurochemicals that are absolutely central to your ability to focus. If you understand these, you understand focus. The first is acetylcholine. Acetylcholine is the neurochemical of spotlight attention. When you decide to pay attention to something, genuinely commit your cognitive resources to it, neurons in the basal forebrain release acetylcholine. It then travels to the cortex and acts like a chemical highlighter. It marks certain neural circuits as important. It says, remember this, wire this deeper. This is the direct molecular link between focus and neuroplasticity. Acetylcholine doesn't just help you concentrate, it signals to your brain that this moment is worth encoding. The more precisely you focus, the more acetylcholine is released. And the more acetylcholine is present during an experience, the more deeply that experience changes your brain. The second is norepinephrine. Norepinephrine, also called noradrenaline, is the neurochemical of alertness and urgency. It's released from a structure called the locus coeruleus, and it does two things relevant to focus. It increases neural signal-to-noise ratio, meaning it makes important signals clearer relative to background noise, and it drives the sense of urgency and motivation that gets you into gear. Low norepinephrine looks like brain fog. Appropriate norepinephrine looks like sharp, alert attention. Too much, and you're in fight or flight, where focus collapses entirely. The third is dopamine. Dopamine is often mischaracterized as the pleasure chemical, It's more accurately described as the anticipation and motivation chemical. Dopamine is what makes you want to move toward a goal, what makes sustained effort feel worthwhile. For focus, dopamine is the engine. Low dopamine and tasks feel impossible to start. Healthy dopamine tone, and you can voluntarily enter deep work states because your brain assigns value to the effort. There's a fascinating interaction between these three. Acetylcholine, norepinephrine, and dopamine don't operate in isolation. They modulate each other, and together they define the neurochemical signature of deep focus. The good news? All three can be influenced through specific behavioral tools. We'll get to those in the protocol section. Now we get to the heart of it. Neuroplasticity is the brain's ability to change its own structure and function in response to experience. It is arguably the most important discovery in neuroscience of the last 50 years, and it fundamentally changes how we think about what learning, skill acquisition, and even identity actually are. The old model was, the brain is fixed. You're born with a certain number of neurons. By adulthood, the architecture is locked. That model is wrong. Your brain is not fixed. It is continuously remodeling itself. Synaptic connections strengthen and weaken. Cortical maps expand and contract. New neurons are generated, particularly in the hippocampus, across your entire lifespan. But here's the crucial point. Neuroplasticity is not passive. The brain doesn't just change randomly. It changes in response to signals that tell it change is needed. And the most powerful of those signals is focused attention. Here's the mechanism. When you focus intensely on something, the acetylcholine released during that focus binds to receptors on the target neurons. This triggers a downstream cascade that ultimately leads to long-term potentiation, the cellular process that strengthens synaptic connections. Put simply, the neurons that fire together during your focused attention begin to wire together more strongly. This is Hebb's rule. Neurons that fire together, wire together. Focus is the trigger for that firing, and the depth of focus determines the strength of the change. This is why passive exposure to information doesn't lead to learning. You can listen to a podcast while distracted. You can read while your mind wanders. But without focused attention, without the acetylcholine signal, no long-term synaptic change occurs. The implications are profound. It means that your capacity to focus is not just a productivity variable. It's the gateway to becoming a different version of yourself. The skills you want, the habits you want to build, the person you want to become. All of it runs through focused attention. Here's one of the most practical and underused tools in the neuroscience of focus, and it costs nothing. Your visual system and your attentional system are not separate things. They're deeply, mechanistically linked. Your eyes have two modes. The first is panoramic vision, a broad, relaxed awareness of the entire visual field. This is associated with a calm, default mode network state in the brain. It's low arousal, low focus. The second is focal vision, a narrow, sharp point of attention directed at one specific target. When you engage focal vision deliberately, you don't just see more clearly. You trigger the neural circuits associated with focused attention throughout the entire brain. The orienting network Posner described, it responds directly to where your eyes are pointing. So here's the technique. And it's backed by neuroscience, not productivity gimmicks. Before a deep work session, pick a single point in your environment. A spot on the wall, the corner of your screen, a single letter on the page in front of you. Spend 30 to 60 seconds staring at that single point. Deliberately, consciously narrow your visual field to just that point. What's happening neurologically? You are manually engaging the orienting network. You are releasing norepinephrine via locus coeruleus activation. You are signaling to your prefrontal cortex that you are transitioning into a focused state. It sounds almost too simple, but the data is clear. Visual focus primes neural focus. This is why athletes enter a stare before a performance, why meditators fix their gaze before sitting, why surgeons visually lock onto the operative field before the first incision. You can use this deliberately, not just intuitively. Try it before your next important work session. You've heard of circadian rhythms, the roughly 24-hour biological clock that controls your sleep-wake cycle. But fewer people know about a shorter cycle that may be even more relevant to daily focus, the ultradian rhythm. Ultradian rhythms run approximately every 90 minutes throughout the day and night. During sleep, they organize your REM and non-REM cycles, but during waking hours, they organize your brain's cycles of high arousal and low arousal. Research from sleep scientist Nathaniel Kleitman, the same scientist who discovered REM sleep, identified what he called the basic rest activity cycle. Roughly every 90 minutes, your brain goes through a full cycle of heightened alertness and cognitive capacity, followed by a trough of lower arousal and reduced focus capacity. Most people fight these troughs with coffee, stimulants, or sheer willpower. That's a mistake. Neurologically, the 90-minute peak window is when your prefrontal cortex is most capable of sustaining top-down, goal-directed attention. The trough is a period of neural consolidation, when your brain is actually processing and integrating the information from the previous peak. Here's the protocol implication. Design your day around 90-minute focused work blocks, followed by genuine 20-minute rest periods. Not scrolling your phone, not checking email, genuine rest, eyes closed, quiet, or a short walk without headphones. This honors the ultradian architecture of your brain. What you'll notice: the first 5 to 15 minutes of any focus session are typically difficult. This is neurologically normal. It takes time for the acetylcholine and norepinephrine systems to fully engage. The reward is the 60 to 75 minutes of genuine deep work that follows. If you abandon tasks at the first sign of difficulty, because the first 10 minutes feel uncomfortable, you're stopping before the focus state even fully activates. Push through that initial friction. It's not a sign that you can't focus. It's a sign that your focus system is warming up. Your brain state going into a focus session is not random. It's determined by your biology in the hours before. Two of the most powerful neurochemical primers are physical exercise and cold exposure. A single bout of aerobic exercise, particularly Zone 2 cardio or short high-intensity work, significantly elevates both norepinephrine and dopamine in the prefrontal cortex for several hours afterward. Research from Harvard psychiatrist John Ratey demonstrated that moderate aerobic exercise produces neural changes comparable to a small dose of attention and mood medication simultaneously, improving focus, mood, and motivation. The mechanism: exercise activates the locus coeruleus directly, driving norepinephrine release. It also triggers BDNF, brain-derived neurotrophic factor, which promotes synaptic growth and supports the plasticity that focused learning depends on. Practical takeaway: even a 10 to 20-minute walk or jog before a focus session meaningfully primes your neurochemistry for sustained attention. Cold exposure, cold showers, cold plunges, or even brief face immersion in cold water produces a dramatic spike in norepinephrine. Studies have shown blood plasma norepinephrine levels can increase by 200 to 300% following cold water immersion. This effect can persist for 2 to 4 hours. This is not about toughness or discomfort for its own sake. It's a deliberate neurochemical tool. The cold-induced norepinephrine surge sharpens the signal-to-noise ratio in your cortex, the same mechanism that makes you feel unusually clear-headed and alert after a cold shower. Used strategically before a deep work session, cold exposure can be one of the fastest ways to shift your brain into a high-focus neurochemical state. Here's a counterintuitive one. Boredom isn't the enemy of focus. It may be one of the most important tools for building it. When your brain has nothing external to process, it activates what neuroscientists call the default mode network, a set of interconnected regions including the medial prefrontal cortex, the posterior cingulate cortex, and the hippocampus. The default mode network is where your brain does something remarkable. It connects disparate ideas, it consolidates memories, it simulates future scenarios, it generates creative insight, it processes unresolved emotional experiences. In a world of constant stimulation, phones, notifications, ambient media, we are systematically starving the default mode network of the input-free time it needs to function. And here's the focus connection. Research from neuroscientist Jonathan Schooler and others has shown that regular mind-wandering, true, device-free, unstructured mental time actually strengthens the capacity for deliberate focus when needed. The brain, like a muscle, requires contrast, deep effort and deep rest, focused engagement and open, wandering thought. The practical tool is this: deliberately schedule periods of boredom. No phone, no podcast, just walking, sitting, showering. With your thoughts, 10 to 20 minutes daily. This is not wasted time. It is cognitive maintenance. Your prefrontal cortex strengthens its top-down control of attention when it is given the opportunity to experience contrast. Periods of deliberate engagement, followed by periods of open, unconstrained processing. Let's bring this all together into a concrete, practical protocol. 5 steps, science-backed, ready to implement tomorrow. Step 1. Neurochemical priming in the morning. Within 90 minutes of waking, do either brief aerobic exercise, 15 to 20 minutes, or cold exposure. This elevates norepinephrine and dopamine before your first focus session. Delay caffeine for 90 to 120 minutes after waking to allow adenosine to clear naturally. This prevents the afternoon energy crash most people experience. Step 2. Visual focus reset before each session. Before beginning any deep work block, spend 30 to 60 seconds in the visual focus technique. Lock your gaze on a single point. Breathe slowly. Deliberately engage your orienting network and signal to your prefrontal cortex that a focus state is beginning. Step 3. 90-minute focus blocks. Work in single-task blocks of up to 90 minutes. No phone, no browser tabs unrelated to the task. Expect the first 10 to 15 minutes to feel difficult. This is your focus system warming up, not a sign of failure. Once in state, protect that state. Step 4. Genuine rest between blocks. After each 90-minute block, take a genuine 20-minute rest. No screens, eyes closed, a short walk, or simply sitting quietly. This honors the Ultradian architecture of your brain and allows default mode consolidation. Step 5. Scheduled boredom. At least once per day, spend 10 to 20 minutes with nothing to do. No input, no task. Let your mind wander. This is default mode network restoration and is non-negotiable if you want to maintain long-term focus capacity. Run this protocol consistently for 2 to 4 weeks. Given what we know about neuroplasticity and the role of focused attention in driving it, consistent practice will not just improve your focus in the short term, it will physically change the architecture of your brain over time. Focus is not a personality trait. It is not a fixed limit. It is a biological capacity built on neurochemicals, driven by specific circuits, and changeable through deliberate practice. Every time you choose to sit with a difficult task instead of reaching for your phone, you are releasing acetylcholine. Every time you push through the uncomfortable first 10 minutes of a work session, you are training your prefrontal cortex. Every time you rest properly between focus blocks, you are honoring the Ultradian architecture your biology was built on. You are not just getting more done. You are literally building a different brain. That's not motivation. That's neuroscience. This has been NeuroWise, science for your everyday brain. If this episode gave you something useful, subscribe so you don't miss the next one. Leave a comment with the protocol step you're going to try first, and share this with someone whose brain deserves the upgrade. We'll see you in the next one.